Okay, so it is the 19th of April. So we've talked about, Mendelian monogenic traits. We've talked about traits that are inherited a little bit differently, where they're either codominant or there's incomplete dominance. So now we're going to talk about some other traits that are Mendelian, but basically something very strange that was observed at some point. Is it hard to take me seriously with purple hands? So Bateson was one geneticist, and Punnett, you might have recognized that name. Punnett was another one. And this is in the early 1900s. They were basically trying to reconfirm some of Mendel's work with the pea crosses. And they were using plants called sweet peas. And these are sweet peas right here. They are a beautiful plant. You can grow them in your garden. If you bought seeds in the Science Club seed sale, we were selling sweet peas this year. You could have planted red and purple sweet peas. Cool. So, turns out flower color in sweet peas is monogenic. It is Mendelian. It is controlled by a single trait. Well, pollen shape is also Mendelian in sweet peas. Single trait, monogenic, simple inheritance pattern, dominant recessive. Great. And the two options for flower color are red and purple. And the two options for pollen, some of the pollen grains are sort of long, like cylinder shaped, and some of them are really perfect little beach balls. Okay. So they were crossing heterozygous individuals. How do you get individuals that you know are heterozygous? Well, what if I take two individuals that I absolutely positively know for certain are big P, and I cross that individual with an individual who I know is, little p. What do I get? I get all heterozygous offspring. And this is convenient because then you can take your heterozygous offspring, you can cross them. And they did this, and they studied inheritance patterns. And guess what pattern they found in a monohybrid cross? 1 to 2 to 1, 3 to 1 phenotype. Well, then do you remember the dihybrid squares, where we cross everything together? Well, they did a dihybrid square. And remember, when we do a dihybrid square, we have this parent. And when they manufacture gametes, we expect that we'll see big P, big L, big P, little p, big L, little p, little l. We do the dihybrid Punnett square. What do we get for a ratio? Do you remember? Let me get rid of this. That's the genotype ratio. Good. So I didn't specify, did I? So if we look at the phenotype ratio, what are the possible phenotype combinations in a dihybrid cross here? Well, I'm glad you asked. We could get purple flowers with long pollen, right? Dominant. We could get purple flowers and round pollen. Whoops. We could get red flowers, which are recessive, with long pollen grains, which are dominant. And we could also get red flowers with round pollen grains. And let me shrink this and move it around a little bit. So what is that usual phenotype ratio in a dihybrid cross? 9, remember this? 9 to 3 to 3 to 1. So they expected that if they got just 16 individuals, they would have 9 and 3 and 3 and 1. Well, you don't breed 16 individuals when you're doing genetic research. You breed 100 or 1,000 or 10,000. You cross these individuals until you get really big numbers. So if we remember, 9% is about 56%. So if they got 100 individuals, we'll say n equals 100, they expected that they would see about 56 purple, and I think it's about almost 20%, like 19%. And then this last one I think is about 13%. So, okay. And, if they had gotten like 58 and 21 and 17 and 10 with 100 individuals, they'd say, yeah, we're in the right ballpark. This is good. Why are we talking about this? Well, because they didn't get what they expected. So what they got was more like, so here's expected, and here's what they actually got out of 100 individuals. It was more like 72 and 3 and 5 and 28. I didn't make those numbers add up perfectly. Whoa! That is different than what they expected. And whenever we get something that is different than what we expect, that's when we kind of sit up and go, okay, hold on, I don't understand what's going on here. And that's what they said. So they reran the experiment. They recrossed these heterozygous individuals. Guess what? Happened again. Oh. Well, that's weird. So it's almost as though the dominant gene for flower color and the dominant gene for pollen are somehow linked together. They almost always go together. It's almost as though the recessive gene for flower color and the recessive gene for pollen shape are somehow linked together, like they always go together. Have you ever observed any other traits in any species that remind you of this? What do you think? Okay, we see more light eyes with light hair than we do light eyes with dark hair. Huh. We see more redheads with blue eyes than we see redheads with brown eyes. Huh. Maybe we can have people with black hair and blue eyes. One of my best friends, black hair, blue eyes. That happens. But very often, coloration goes together. Now, hair color, eye color, skin tone in humans, controlled by more than one gene, what do we call that? Polygenic, right. So it's not a perfect example. These are monogenic. These are controlled by a single gene. But it seemed like they were linked together somehow. So, do you all have your chromosomes somewhere in your notebook? Get out your chromosomes. Okay, so you remember that, how many pairs of chromosomes do you have? 23. You have 23 chromosomes. Yeah. Now, in your hand, you should have 2. And you got one copy of each of those chromosomes from your mother, one copy from your father, one's a maternal copy, one's a paternal copy. And when they get divided up and go off to eggs, they do this weird thing first. They wrap their little arms and legs around one another. So it looks a little bit like this. So they sort of line up side by side before they go off to their respective gametes, their respective eggs and sperm. And the bottom of the chromosome actually twists around one another. You can do this with yours if you want. And the weird part is what very often happens next. The ends break off and they switch. They switch. And so what you end up is with magic. I did it by magic. This happens in your cells every time you produce gametes. So it mixes up traits. You do not have to do this with yours. Please don't try, because yours are all tightly wound. These are just sort of a fake pair that I did for show. But then what we get is these 2 copies, where they've traded ends and now they can go off on their own. And what this does is it mixes up traits. So it means you can have, you can pass on your nose and your partner's eyes, or your crooked pinkies and your partner's not connected earlobes. Well, what is a gene on this chromosome? What is an actual gene? Hey, focus, Pinky. What's an actual gene? Is it bigger than a chromosome, smaller than a chromosome, on a chromosome? Where is it? It's on a chromosome. It's actually a section of that DNA, remember? So it might be from here to here on that DNA. Well, here's the thing. If you have two chromosomes, or two genes on the same chromosome that are side by side, they hardly ever get mixed up. They tend to travel together. And so that is what we call gene linkage. So here's what they found. That basically the dominant traits for flower color and pollen shape were traveling together all the time. Because if you look back at their Punnett squares, there were way more of those purple long than you'd expect. There were also more red short than you would expect. There were not nearly as many dominant recessive combinations where you had purple round or red long, because it seemed like the genes for those two traits were side by side, so they don't get split up very often. So the dominant genes tend to travel together, and the recessive genes tend to travel together. Now, a couple words about gene linkages. Gene linkages can only happen if two genes are on the same chromosome. They can't ever happen if two genes are on different chromosomes. And they tend to not happen when the genes are far apart on a chromosome. So you tend to see gene linkage when the two genes are near one another on the chromosome. The closer they are, the more strongly linked they are. So the closer they are, the less frequently you'll ever see a recessive dominant combination. So the big idea is when you have a dihybrid cross between two heterozygous individuals, if there is no linkage between the genes, like if they said these two characteristics are, these two traits are on separate chromosomes, okay, no gene linkage. Good, 9 to 3 to 3 to 1. If there is a gene linkage, if they tell you, for instance, these two genes travel, they're side by side on the same chromosome, you would start to think, oh, they're probably linked. Then if they ask you for a phenotype ratio, you need to deviate from 9 to 3 to 3 to 1. So that's the whole key. And they may not even say gene linkage. They may say, two genes which are carried on separate chromosomes. Then you have to mentally go, oh, no linkage. Or if they say two genes that are side by side on chromosome number, whatever, 32. We don't have 32 chromosomes. Carrots do. Carrots have 2,000 and some. Then you start to think, oh, there might be a linkage. Okay. I'm going to read that again. Eye color and fruit wing size and fruit flies are monogenic traits located side by side on chromosome 2. Red eyes, big R, dominant. Green eyes, little r, recessive. Long wings, big L. Short wings, little l. If we cross two heterozygous individuals, and you had to show phenotype ratios. And this is pretty much exactly like the question that was on the test last year. What would it be? Okay, so what they're asking you to do is put in percentages on this bar graph for the phenotypes. And I'm actually going to move this just a little bit more, because I want these percentages labeled. And this is pretty close to exactly what was on the air test last year to address this standard. Okay. You put in bars for what you feel would be appropriate. So if it was 9 to 3 to 3 to 1, what would it be? Like 56, 20, 20, and 13, I think. That's what we figured out. So if you want to say that red long, which is the dominant phenotype, correct, to get points for that answer, what do you think you need to put in there? More than 56%? Less than 56%? We're saying that these two traits are carried side by side on a chromosome. Would we see the dominant traits running together more often or less often? more often. So if we take the red long, and we could go up to 75% easily. Because we know that it's going to be a significant portion of these that are dominant-dominant. Now, could you probably get credit for an answer that was that? Yeah, probably. You know, is it likely that it's going to be 60, 65, 70%? Yeah. Again, there aren't perfect answers for this. If you put in a little bit of that, and then something like that, you're showing that, you know, roughly a quarter of them are recessive, nearly 3/4 of them are dominant, and you've got a few that get a mix. But for the most part, it tends to cluster here and here, and that's what they want you to show. Okay. Now, if they say that these two genes are on different chromosomes, does that change what your answer should be? Yeah. Then you should be putting in, you know, 56, 20, 20, 10, something like that, getting them to add to 100. Then you're looking back at your 9 to 3 to 3 to 1. But if they're telling you that the genes are side by side on a chromosome, then you know that you have to be looking at deviating from that 9 to 3 to 3 to 1. Okay, questions, comments?